Welcome to Applied Stem Cell's ongoing webinar series highlighting our technology and how it's being used. I'm Pia Abola, Director of Marketing at Applied Stem Cell, and I'll be your moderator. Today, we'll hear from our collaborator, Larry Forman, founder and CEO of CHO Plus, a company focused on increasing the productivity of CHO and HEK cell lines for advanced therapeutic production. A biologist by training, Larry has academic and industrial experience that includes mammalian cell culture process development at Genentech and other companies. In 2014, Larry founded CHO Plus to finally and successfully develop cell engineering methods to significantly increase the productivity of mammalian cells for therapeutic protein manufacturing, viral vector manufacturing, and other purposes. Today, Larry will be speaking about the work that CHO Plus has done to optimize production of AAV and HEK293 cells and therapeutic proteins in CHO cells. Before he begins, I'd like to invite you to use the Q&A panel to ask questions, and I will present them to Larry at the end of his talk. With that, I'd like to turn things over to Larry.
Well, thanks very much. I appreciate the invitation and the opportunity to speak with these folks. I'm going to take you on an interesting journey. A lot of information will whiz by. I think you'll pick up a lot, and I'm happy to answer questions either at the end or offline. So let's begin. I guess let's start by asking, can everyone see my screen? Okay, great, thank you. Okay, so why is the company called CHO Plus? Well, the first thing we did was engineer CHO cells to increase therapeutic protein, particularly antibody production. A note to all of you, if you feel like starting a company of your own someday, don't name it something specific, but we'll get to that. So here's the problem, that factories to produce therapeutic proteins are very costly. Some cost as much as $2 billion each. There are other issues, for example, continuing capacity limitations, because every year there are more and more therapeutics approved, and then those that have been approved find new patient audiences and their expanded uses. So the industry's overall reaction to this has been to simply build more factories as they go. Also, I want to point out that I've been in this business since the 1980s, and actually the current state of affairs is an average productivity for therapeutic antibodies of about 4 grams per liter. Some people do better than that, some not as well. There's a little background noise, so I apologize for that. So here's the issue that many people have observed. On the x-axis, you have message copy number or gene copy number. They're certainly related. And on the y-axis, you have specific productivity. And what everyone has observed is that initially with low copy number of message or gene of interest, you get a fairly rapid increase in specific productivity as you increase the copy number. But eventually, you get an arithmetic addition, and eventually you get very little increase in specific productivity. The cells just don't have the capacity to do much more than that, and you've hit the upper limit. Well, so we came up with a cell engineering technology. It's been patented, and it's a directed evolution in a way. Directed evolution covers a lot of different techniques. So we start with host cells. We do repeated cell fusions over and over and over again. And what we do is we create a library of cells with shuffled genomes and randomly amplified whole chromosomes. The next step is rather passive, just looking for good growth. So if during this engineering process, we've killed some cells, that doesn't matter. Or if they're slow growers, we don't want them anyway, and they get diluted out. The next step is where all the action is. So we can do one of two things. We could either apply selective pressure, as we do to get cells which grow faster, or we could screen the library for phenotypes that are of interest. So, for example, endoplasmic reticulum. And here you see cells stained. This is not GFP. That's the first instinct. These are cells stained with green tracker dye for endoplasmic reticulum. On the left, you have CHO-K1 before engineering, and on the right, you have a pool of engineered cells after engineering and screening for high ER. The cells on the right have 10 times more ER per cell on average than the cells on the left, and we have engineered cells to have up to 40 times more ER per cell. So in this case, we would expect this to happen. I showed you this plot just a little while ago. So if we have relieved the bottleneck and increased the amount of cellular machinery to produce recombinant proteins, then when we increase the copy number of the gene of interest or message copy number, then we should see specific productivity continuing upward. And this is what we've observed. Well, assuming that this is the case, then you might go from about 4 grams per liter average, and we project up to 20 grams per liter and maybe even more. And if this is the case, and if that we could get very wide distribution and acceptance of our technology, and we're working on that, well, that means maybe 80% of these very expensive manufacturing facilities don't need to be built in the future, since you have much higher productivity for existing or new facilities. So let me show you some results. And let me first tell you a little about the format of the results. So here we have stable transfections of an IgG1 antibody. And in the left panel, we've got a comparison between the parent pre-engineered versus a pool of engineered cells. I want to emphasize that it's a pool of engineered cells, and we expect a lot of diversity. It's not really a pool that's a transfected pool where you transfect into a cloned parent. So we have wide diversity in this engineered pool, as I mentioned earlier. And so here we see about a 10-fold increase of average specific productivity for the engineered pool versus the parent cells. And when we look at volumetric productivity, admittedly, the parent cells are producing rather low, but we see about a 10-fold difference. And then on the right, here's where things get pretty interesting. We use Sleeping Beauty transposase to do our transfections, and many people are using transposases these days. Well, what people who use transposases for transfection have come to accept and to expect is what you see for the parent host plus antibody gene of interest. And that is a pretty narrow distribution around the average for this transfected pool. However, remember, we're transfecting into a very diverse pool of engineered cells. And so our average is much higher. That's marked there, I think, by around there. But because we have this wide diversity of karyotypes, we see some extremely high producers, and aside from a few stragglers, almost all of them are at least as good as the average for the pre-engineered cells. And we'll talk about this in a little bit more. So I want to buzz through some of these things and leave you with concepts more than specifics. So when we look at specific productivity, we've achieved up to 117 picograms per cell per day, measured very conservatively, with volumetric concentrations of up to about 13.5 grams per liter. Oh, that's not shown on this one, but we have achieved up to 13.5 grams per liter in shake flasks. And we see we could do a little better on medium development, but we see pretty similar growth curves in general. We could tease that apart another time. I think here we have the higher concentration of 13.5 grams per liter, and somewhat better growth. So this is only with about 20 million cells per mil peak, and so this is a pretty good value. We would expect this to carry over into large-scale bioreactors too, and we will be doing that in the coming months. Regarding product quality, we have taken a look at several things, and what we see is that with higher productivity, people expect more aggregation. We don't see that. And actually, we see less product aggregation with a bispecific, difficult to produce antibody using our engineered cells. We expect different clones to have different glycoform profiles, and with several or many different high producers, we can pick and choose those with the desirable glycoform profile, for example, for biosimilars, or for a licensed product where you're changing the host. However, if you're starting with a new product, it's a little bit less important because you don't have to match a glycoform profile. You just need consistency in the lifetime of the product, and that's not a problem. Another thing that I'm not showing, but we can do is we can also engineer cells for higher levels of Golgi apparatus, and we think we could improve and increase the sialic acid site occupancy, again, for new products and not for biosimilars. Okay, so cell line stability is important, and people have raised these questions, and it makes sense. So, is there an effect of high productivity on growth? And the answer is no, we haven't seen a negative effect on growth, but you need some more nutrient feeds, as you might expect sometimes. People also thought, well, the high productivity may have an effect on production stability. And in fact, and I'll show that on the next slide, that we don't see this, that 83% of our clones are stable for at least 60 generations, which is sort of above the industry average or expectation. We have not looked directly at genetic or genome stability. However, because we have stable production over 60 generations, that implies that the genome is also stable, but to be explored further in the future. We also have seen that engineered cells seem to maintain the high ER phenotype for at least 6 months. So we're in good shape there. So this is just to show you that we've done 60 generation stability. Out of 12 clones that we looked at, only 2, this one and this one, were not stable. And interestingly enough, one of the unstable ones actually increased. And when we have time, we'll explore why this happened. I can guess, but that's beyond the scope of today's discussion. So not bad, 10 out of 12 stable. That's actually very good. So what else can we do with our engineering platform for CHO cells? And one thing we can do is we can increase or we could increase the growth rate. So what we do is we take our engineered pool of cells and we apply selective pressure. And this selective pressure is in the form of more frequent passaging and longer dilution on each passage. And what we can do is isolate clones that have doubling times about 50% faster than the starting cells. So we start with doubling times of around 22 hours, and we end up with cells with doubling times of around 13.5 hours. So that's also a pretty interesting feat, considering many people have worked a long time to increase growth rates, and they don't expect cells with a larger genome to grow faster, but in this case, they do. We've achieved up to 13.5 grams per liter in shake flask for an IgG1, up to 117 picograms per cell per day, 60 generation stability, less aggregation, and we have not really done anything with glycoform analysis yet. So let's switch gears to viral vectors. And I'm really a protein guy, and I've worked in process development for therapeutic proteins for a long time. I don't want to say exactly how much, but I'm a quick study, and I've explored the viral vector space. So the first thing that I learned is that there are many different approaches to produce viral vectors, such as AAV, but really the outcomes are not very different, even though people use different methods, different leader sequences, different vectors, different promoters, etc. And really a lot of improvement is needed. Now, this is worth spending a few moments on because it tells you really what some of the problems in this space are. So we all know about the treatments for very rare genetic disorders that cost anywhere from $2 to $3 million per treatment or more. And by the way, those treatments are not profitable. Well, let's just say $2 million for now. And the other problem is that some of these treatments may require 10 up to 40 or 50 liters of production medium to make enough viral vectors to treat a single patient. But again, let's be conservative and say 10 liters, and let's do some math here. Let's assume that you have 1 million patients you want to treat, maybe not for one disorder, but for a handful of more common disorders, or for non-genetic diseases, but diseases that can be treated with a genetic therapy. Things like heart disease, where you're using a gene therapy to improve the outcome. Well, if you have $2 million per patient and you have 1 million patients, that's $2 trillion, and that's more than twice the US defense budget. And there's just no one who's going to pay that. And at 10 liters per patient, that's 10 million liters. So that's either 520,000 liter bioreactor cultures, and actually triple transient transfection hasn't been scaled up much above 500 liters. Or you could have 20,500 liter bioreactor cultures, and you could see that's not going to work either. So these are some of the problems with low productivity. And they're all related except for the last one: low productivity, low percent full, the high cost, and the inability to produce enough viral vectors for more common disorders. And the final product quality is also an issue with a sort of, I wouldn't say it's a standard, but it's common to have about 80% full. And certainly the higher the percent full, the better for the patient. Immunogenicity is really related to the nature of the capsid and the targeting that's desired. So we asked the question, can HEK293 cells be improved by a directed evolution in similar fashion to what we do for CHO cells? And of course, you could guess the answer is yes. So we do our repeated cell fusions, and then we look for different phenotypes. So not to go into too much details here, because endoplasmic reticulum is only important really for protein productivity, not for viral vector production. But mitochondria, while important for both, is really very crucial for increasing virus productivity and increasing percent full. Not only increasing the mitochondria per cell, but also high reactive oxygen species and high membrane potential. So we've chosen to work on 4 different serotypes: AAV1, 2, 5, and 9. Together, they make up about 2/3 of the AAV serotypes that are currently in the clinic. So it's a pretty good effort. We can't do all of them, but we're willing to work with others on them. And for some of the initial experiments, we use the very standard triple transfection method with 3 vectors, one for the transgene, one for rep-cap, one for the helper. And these are some of our initial results. Let me just guide you through what the 3 bars represent. So in our set of 3 bars, the first is our parent cells, unengineered. The second is a pool of engineered cells. And the third are clones taken out of those pools of engineered cells. So actually, I'll just point out that all the engineered pools are the same here. So they all have, they've all been engineered with randomly increased chromosomes, and they all have high mitochondria, et cetera. But when we look at clones, and we expect a lot of diversity because of the many different, maybe hundreds of thousands of different karyotypes. So clones are going to be, they're not going to be tightly situated around the average for the pool. So we see at our best about 4 times 10 to the 16 viral genomes per liter. You see that for AAV2 in the middle there, or about 20-fold higher than the unengineered parent. And we see good results also for AAV1 and 5. Now, a problem that people have observed is that when they do something to increase productivity, they often see a decrease in the percent full. But what I want to show here is that we don't see that our percent full is comparable. However, we can engineer cells specifically looking to increase the percent full, as opposed to simply looking for higher productivity while maintaining the percent full. So Here you have the same situation with 3 bars for AAV 1, 2, 5, and 9. And what we see is approximately a 2-fold increase in percent full is possible. And we're really not putting a lot of effort into this yet until we have partners for this. So we're doing remarkably well considering that we're not putting full-time effort into this. And our peak here is for AAV5 at about 55% full before purification. And we don't have any doubts that for AAV5 and other serotypes that we might be able to get to 80% full before purification. So what are some of the other capabilities? And we are working on packaging and producer cells, as are many other people. And I'll leave this method slide, but now we only have pairs of bars instead of 3 because we haven't had the time to clone these out. So we're comparing parent with engineered pool, and there's no doubt that if we were to clone out the pools that we'd find much better results yet. So when we have tight control over stably transfected rep, cap, and helper, and only our gene of interest is transiently transfected. So we see pretty good productivity. A lot of people who've done packaging and producer cells have seen very big loss of productivity on doing that. And even for our engineered pools, we get good results without much loss, and we expect even better results when we have the chance to clone these out. With regard to percent full, our results are not quite as good as what we did before with our engineered cells with triple transient, but still excellent, and in round numbers, up to 2-fold increase in percent full for our packaging cell lines. So another summary I'll have to skip. People ask about stability, and in this case, we had some cells with the helper element stably transfected and using Sleeping Beauty transposase, and we see 60 generation stability for that. Again, for the sake of brevity, I'm going to move quickly. What we did was a 2-step purification for some of those cells from the AAV5 cultures. And by starting with higher amounts of viral vector and a higher percent full, we get a much improved percent yield recovery, and at the end, a higher percent full of full capsids. So instead, for example, the percent full is very important, and while for the unengineered cells, we got up to 80%, we got up to 90% full capsids since we started from 55% full, and we think we could get to even much higher, 95% or higher percent full, if we can get the productivity and the percent full even higher. So this is what's going on with percent full. And actually, percent full is more important economically than simply increasing productivity without increasing the percent full. So the current state of affairs is on the left. It's very common to get about 20% full and 80% empty. And what you'd like to achieve after purification is about 80% full and 20% empty, but you lose a lot of your full along the way because you don't have baseline resolution when you do separations. Now, on the right is what we are thinking about. If we could get to 80% full before purification, it would not be difficult to get to 95% full after purification and without very much loss of productivity or loss of the full capsids, I should say. So we have some new results, and this is what we're doing in collaboration with Applied StemCell, where we're using their TARGATT landing pad system along with some of our engineered cells, and we're just putting the pieces together, so we haven't gotten too far along, but I wanted to share some of these results with you. So the objective is to first insert landing pads into the high transcription region of locus H11. And our thought is that if we did this with our engineered packaging cells that already have other elements stably transfected into them, that we might be able to go from a packaging cell line and do a stable transfection with our gene of interest into the H11 locus using the TARGATT system and come up with clones of producer cells within a few weeks, maybe a month or less. And this would be big because right now a lot of people go into the clinic with triple transient transfection, and then they think that they will switch over to a packaging or producer cell line between phase 2 and phase 3 clinical trials. So what we're doing is using EGFP and mCherry, and I'm going to buzz through this quickly. What we see is the production of both products, the mCherry and the EGFP payload. And what we see here is for AAV 5 and 2 that we get, these are with unengineered cells. But what we see is when we transfect into the H11 locus, we get much higher productivity than we do random integration. So this is a good sign. And as I implied before, we're putting together a lot of the nuts and bolts and pieces that we will eventually assemble into fully integrated and inducible packaging and producer cell lines. So what our engineering approach does is increases the capacity of the cells or reducing the bottlenecks in the cells to do more of what they already do. So, and that's what we're doing. We're not engineering in any new features, but if the cells can do something, if they could perform some useful function, we could improve upon that. And we've shown that in so many ways. All of our engineered cells, both CHO and HEK, will meet regulatory agency regulations. We don't use any foreign genes or viruses. They're all suspension adapted, and we use commercially available medium, serum-free, animal-free, and chemically defined. And we're just working in shake flasks right now. You saw this slide before. Again, all except for the last are related, and we're in the process of addressing all of these. Immunogenicity really still depends on what kind of capsid is desired. I spoke about some of this, but basically we're always looking to improve with higher productivity or percent full. We're working towards fully integrated, stable inducible producer cells. We're continuing our work with the TARGATT landing pad system, and we hope to generate a new standard of 95% full post-purification. Well, we have 4 lab scientists, but all the work that I presented today was performed by Kathy No. And thank you all for listening. I think I ran a little longer, but I had a lot to talk about with CHO as well as HEK cells. So happy to answer questions if anyone can stay around. I hope you can.
Thank you very much, Larry. Really appreciate sharing your the insights and your findings. I actually have quite a few questions for you. One of them is I'm curious about the percent full capsid and why does your system able to give much higher percent full compared to what everybody else is doing? Do you have any insight on that?
Yes and no. We couldn't predict exactly what phenotypes of engineered cells are going to be important. What we're doing is we're developing a toolbox of cells with high or low mitochondria, or high or low membrane potential, or high or low reactive oxygen species. And there are several other phenotypes that are beyond the scope of today's discussion. So we're developing cells that have collections or constellations of these different phenotypes. It's not infinite, and we can test different serotypes with these different clones and find the ones that are best suited for that particular serotype. So it probably would be very difficult for an AI or machine learning system to predict exactly what combination of phenotypes is going to be best for a particular serotype. But it's not random, but we can't predict either. So what we're doing is we're developing cells with additional capacity and abilities, and we're figuring out ways to find them quickly.
Thank you. I'm curious if that is, like, my next question may be related to that. Earlier in the talk, you mentioned that you were overproducing, getting really high titers of proteins, and they weren't aggregating. How come, if you have so many more, why don't they just glom together
Well, that's a really good
or
question too.
unfold? The protein folding, in the ER.
Yeah. Well, we can just guess, but it's probably a good guess. Normally, aggregation takes place extracellularly, and it's depending on medium pH, ionic strength, or medium components. But in the case of the difficult to produce protein, it was pretty clear from our partner that the aggregation was occurring before it was secreted.
Mm-hmm.
So what we could probably say is that there's something about the intracellular conditions. It may be that the secretion machinery was working quickly enough um, to secrete the protein before it had the chance to aggregate. That's number one. Number two, some of these cells might have had slightly different intracellular pH or different intracellular redox potentials that might have helped. So uh, I I couldn't know specifically, but probably something along those lines, because when we look for cells with high amounts of endoplasmic reticulum, we already know that's a surrogate, but we already know that there are many other features that are changing, although we may not be monitoring all of them. Different levels of tRNAs, different levels of ribosomes, different levels of Golgi, which we can but have not been monitoring lately, and different levels of chaperones and other secretory machinery.
Mm-hmm.
So we're making for narrower and narrower haystacks with phenotypes that we know, and then there may be some that we don't know. And when I say phenotypes, these are not new phenotypes, since all the chromosomes and all the genes are, in the case of CHO, they're all CHO, but different ratios. Okay, so some are elevated, some are normal, and some are in between. And that combination, which is hard to predict, will give us the best outcome.
I have another question, actually. So apparently had 4 questions. Another question has to do with, and it might be my ignorance of how this process works, but when does the fusion happen? Does it happen, like, do you have to do the fusion before growth every time and then select, or do you do directed evolution after you fuse everything, fuse cells?
Well, it's a process. So
Okay.
we we start by fusing the cells, and then we let them rest. And some cells are going to die; they're going to throw off chromosomes or whatever. Then we fuse them again. We let them rest and grow, and then we do it again and again. So we're generating hundreds of thousands of different possible karyotypes.
Mm.
Okay, and then we have our engineered pool, and now we could go screen or select for the phenotypes that we want. So they all have endoplasmic reticulum, but we want the cells that have the most endoplasmic reticulum. So we stain them with green tracker dye, we run them through a cell sorter, and we collect the cells with the highest amount of ER per cell.
And that is stably maintained once you've done that?
Yes. So we know from past experience and experience of others that um, all cells can stably maintain more than their normal chromosome number to some point. Actually, CHO cells can maintain about twice the genome size stably as normal, but we depend on it after we do many fusions of going above that number and having them randomly throw off chromosomes, but eventually stabilizing at about twice the genome size, but with all the different permutations and combinations of individual chromosomes.
And then another question is, because they're fused and they have all this ER, are they actually bigger in size?
Well, that's a good question that comes up a lot. And actually, no. Even though we fuse them and they have much more ER per cell, up to 40 times more, they are not bigger. And the next question is, well, why not?
Yeah.
And the reason is that CHO cells, though they're the workhorse of biomanufacturing, actually are pretty deficient in ER in the first place. if you were to compare normal CHO cells with, say, B cells, which their mission is to secrete proteins, those are just full of ER. And CHO cells are pretty deficient in ER in the first place, so there's a lot of cytosol to fill without having the cells actually increase in size.
Okay, well, I think that's it for the questions. Really appreciate you taking the time to share your data and share your wonderful way of that you're going to be using TARGATT, large knock-in platform. So thank you very much.
Sure, it was my pleasure, and we like collaborating with Applied Stem Cell, and look forward to other seminars and maybe one on TARGATT specifically when we have more results to share.
Wonderful. Thank you very much.
Okay, thanks. Have a good day, everyone.
Goodbye,
Bye now.
everyone.